Object salience, as you probably most of you know, is a well-studied phenomenon in visual sciences, especially low-level visual features such as color or size, for example, are among the features that have been studied well, make an object stand out, and it draws your attention automatically. But the salience that I'm going to discuss today, and I've been interested in the past 7 or 8 years now, is not necessarily due to low-level visual features, but it's type of a learned salience, right? So this particular piece of paper is not, more artsy than other things that you see around you, but it has high value, and it grabs your attention. It is well known, for example, that novel objects. So this kid might have many toys, but he may be attracted to this remote control because it's novel. And also, there's some objects that are dangerous, even though like this tarantula is kind of well camouflaged in its surrounding, but it's still detected by you, and it arouses strong automatic responses. This is the salience that I'm calling acquired salience or learned salience. So there's nothing in these objects per se that makes them stand out from their background necessarily or other objects. It is the history that you have with these objects. And we've shown previously that there are multiple dimensions to this acquired salience, including appetitive experiences, and perceptual experience, novelty, familiarity, and aversive experiences. And most of these learned experiences tend to enhance the salience of the objects. The aversive experience has a kind of a bivalent effect on salience. I don't have time to get into it, but sometimes it increases salience, sometimes it decreases salience. Today, what I'm going to focus on mainly is value-based salience, but really we have been exploring other dimensions of acquired salience, such as perceptual-based salience and aversive-based salience. And if I get time, I don't think 40 minutes is enough for me to get into that, but maybe during the panel or after that, if anyone is interested, we can discuss. And basically what I will show you today first is I will give you a what I call a practical or working definition of what I mean by salience. Most of the time when people talk about salience and they do experiments with, it's more like an intuitive intuition about salience. So you have intuition that some object should be salient or not, but normally they don't quantify it. How much an object is quantifiable? The salience of an object is quantifiable. And for that we have a task using free viewing, a gaze bias. So we have a direct quantitative handle on how much salience we've induced in the objects. I will show you that. Then I will discuss some of the about to be published work and comparison of cortex, PFC, and substantia nigra reticulata on value coding, and how it's actually surprisingly happened to be very, very similar in both of these structures. I will also show some again to be published results on the fact that this value-based salience is very granular, means that it has high resolution. So if you add the value associated with an object by a small amount, it can increase its salience by that amount, and it will be reflected in the firing of the neurons. We also uncover a salience related to uncertainty. So not only value itself makes an object salient, but an uncertainty about value has an additional boost to salience. I will talk about that. Next, I will discuss some published work and some recent advances on the fact that we discovered that with long-term value association, you can create what we call efficient search. People who are familiar with visual search, we know that there are two different types of search, or at least a spectrum of searches, from going from serial, slow to parallel. And what we've shown previously is that value can create efficient search, parallel search, and we've started to investigate the mechanisms of that using both modeling and neural recording. I will show you some of the recent data from my lab that have looked into this, and I will also show you results from whole brain imaging in both macaques and humans, trying to find the neural signatures of value-based salience. As for perceptual salience and aversive salience, I guess we have to leave it for another time. I have to also do a cautionary note in the beginning that the value-based salience that I'm talking about is kind of different from what the majority of field is talking about. And mostly when people talk about value-based salience, they mean trial to trial value association to the objects, creating salience. And this type of trial object reward association can be very transient, so it can change in different contexts. It's very flexible. Like, for example, if you play a game of Uno, what is the value of each of these cards? In each trial depends on what card is shown on the deck. So if a card number 5 is shown on the deck, then number 5's in color yellow has value, but the other colors or numbers don't have value. This is what I call transient object salience, and people refer to that in terms of saliency literature as priority. In this particular task, some object has higher priority or lower priority. This is not the salience that I'm talking about. I'm talking about long-term, years-long, stable object reward associations, value associations. So, these paper money that you see on the top have kept value for years or centuries, and the bottom ones have low value. Okay, so stable high value, stable low value. This is the one that I'm interested to look at because it has important behavioral consequences that I will show you during the talk. Okay, so acquired stable object salience. When I talk about value, please don't mix it up with the flexible switches of the value object association. The stimuli that we use in all of our tasks with both humans and monkeys are these fractal objects. They're easy to make. You can make lots of them, and they can have all different sorts of features built into them. So they naturally lend themselves into our research because we don't want to be biased by any low-level features. We want to build associations. So these are abstracts. Monkeys or humans have normally no experience with these shapes, and so we can build these experiences for them during the experiment, and then we can look at the effects. Okay. Now I told you I will give you a quantitative measure of salience. How would I do that? We have developed a task we call free viewing. In this task, we associate objects with some outcomes before free viewing multiple times. Then we show multiple objects at the same time on the screen. This is with macaque monkeys. And monkeys are free to look at these objects. They don't get any reward, any punishments, nothing. It's just that the monkey tends to look at the objects that he, stands out. Behavior has had importance. The salience for him, caused by previous training. Okay, so in this case, you can see these 4 objects on the screen. These are the dots that you see is the saccade trajectory of the monkey, the gaze trajectory of the monkey, going from yellow in the beginning of the trial and then darker blue to the end of the trial. You can see how the monkey completely pays attention to 2 fractals and ignoring 2 other equally beautiful fractals just because objects 2 and 3 have been associated previously and these 2 objects have no reward association. Monkey just likes to look at them. And this is, I'm showing you on the right side, the saccades and the eye location of the monkey. And we've shown not only monkey makes more saccades to these objects, but also scans the details of these objects more. This is the object scanning. These are mini saccades, I call them. They're not as small as microsaccade, not as big as regular saccades, but it's just, looking at the feature, it's as if the monkey is trying to take in as much detail of these objects. He likes these objects and wants to spend time with them. Of course, this is with appetitive objects, but we see similar stuff sometimes with aversive objects as well. So it is really an attention-based mechanism more than the liking that I just described. Okay, so we have a way to measure salience, and we've done this before. So, for example, if you have multiple objects, let's say these 4 objects associated with 3 drops of juice, this 4 with 1 drop of juice, and then in the context of free viewing, you look at the number of first saccades of the objects, how much is scanning, how much viewing duration. All the time you see huge bias for objects with high reward compared to low reward. Not only value itself creates salience, but we've realized that the uncertainty for reward creates additional salience. So these 2 set of objects have equal expected value. Both of the expected value for both of them, let's say, is 2 drop of juice. But this gives you 2 drops certain. This gives you a 50% chance of 1 drop, 50% chance of 3 drops. And in this case, the expected value of these objects that has been learned is the same. Yet, we see monkeys tend to look during free viewing at the uncertain object more strongly, first saccades and object scanning more strongly. Of course, the effects are not as strong as value itself, but it's there and it's significant. Okay, so both value and value uncertainty makes object salience. And the first question that I want to answer in this basically talk is, what is the neural mechanism behind reward-related object salience? And for that, I will basically discuss some of the about to appear results in which we've compared the responses of cortex, prefrontal cortex, and substantia nigra reticulata to these different types of objects. Either we have what we call bad objects and good objects. So bad objects are objects that are associated with one certain drop of juice. Good objects with 3 certain drops of juice. Amount objects are 5 objects in a set with a linearly increasing amount of reward going from 1 to 1.5 to 2 to 2.53. And then we have probability objects that have matched expected value to the amount objects, but probabilistically. So with probability of basically 0 high reward, 25% high reward to 1. So they match the expected value with amount, but they have uncertainty built into them. Okay, so we want to look at how granular is the learning of salience using amount, and we want to see how the uncertainty affects neural firing with probability, and also with good and bad, we want to look at the longevity and the effect. So the task is very similar to the tasks that we do with the monkey, and this is the work that I did in Hikusaka's lab. Where when we ask a monkey to look at the central fixation, an object comes, monkey looks at the object, he gets the reward for that object. Another object, another reward. Different target locations. After the training in few days, we just ask the monkey to simply passively to fixate the center and passively view objects that are flashed kind of rapidly, 400 millisecond on and off, in the receptive field of the neuron, mixing good and bad objects. Okay, we want to see if the neuron can differentiate good and bad objects just by the history. So here in the second task, there's no reward given to the monkey. That's why we call it value memory. So if there's any difference on average for good and bad objects, since we are mixing all the visual features with a large number of objects, over 100 objects for the good and bad, let's say, if there's any difference, that must come from this history of reward tree. Okay. And in this particular task, the neural recordings have been done in 2 regions, not simultaneously, but in the same monkeys and with the same objects. So the same objects, in one session I record in VLPFC, in one session I record in SNR, and these are the regions in SNR and PFC that are known to project to each other, but this is beyond the time for the talk today. So I will show you first examples of the firing of these neurons in one neuron. So this is monkey B. The responses in VLPFC and SNR, the PSCHs, mean responses to good objects in red, the bad objects in blue, and you can see this PFC neuron is highly excited to good objects, stronger excitation to good objects compared to bad objects, and 2 neurons shown with the same object basically. So again, PFC more firing to good than bad, and SNR the opposite. Okay, so this is both of these. But by the way, responses have been reported previously in our previous publications, but comparison of the 2 regions hasn't been done. So here what I'm showing is the distribution of what we call the AUC, so the difference between firing of good versus bad. So if the neuron fires more to good than bad, then the AUCs will be closer to 1. If it fires more to bad than good, then the AUCs will be closer to 0.5, means no difference between the firing to good and bad objects. And you can see in about 300-something neurons in VLPFC and about 100 or something in SNR, the opposite polarity of the average population, even though there are neurons with minority value coding in both regions as well. So in VLPFC, we have some neurons that are what I call bad preferring. They fire more to bad. And in SNR, we have some neurons that are good preferring. They fire more to good. So GP means good preferring, and BP means bad preferring. This is the whole population, and the ones that are colored are the neurons themselves. The AUC is significant, and this is the population. The mean is shifted to the right for PFC, left for the SNR. Okay. Now, one thing that we wanted to know at this point is that which region between these two, this is one of them is cortex, the other one is the output of the basal ganglia. Discriminate the value memories first. So if value memory is reserved or preserved in some region, okay, then that region should discriminate the value of the 2 objects first, and then it can relate it to the other objects. Both of to the other region. Both of these regions are connected to each other. I will show you later a diagram through corticobasal loops. Okay, so we want to know the onset of value signal. And surprisingly, when you put the value signal, so the difference between good and bad firing of SNR and VOPFC on top of each other, so this is for SNR and this is for VOPFC. Of course, SNR signal is much bigger, as you saw in the examples, but when you look at the onsets, there's hardly any difference between how fast VOPFC or SNR is discriminating if an object is good or bad. And you can do a CDF, a cumulative distribution function of the onsets between VOPFC and SNR for all the neurons, and still you don't see any difference, right? I have to tell you that this is a slightly different or unexpected kind of a result, because in another study by Masaharu Yasuda and Hikusaka published previously, they showed that given the fact that SNR inhibits thalamus, if you record in thalamus and SNR and you look at the value signal, this is the difference between good and bad in thalamus and the difference between good and bad in SNR, thalamus response is delayed by about 10 to 15 milliseconds from SNR. So it's kind of consistent with a monosynaptic connection. Now here between SNR and VOPFC, we have at least a disynaptic or triple synapses. They're away from each other. So you expected at least such a big difference, but you don't see that. The onset responses are kind of surprising. And they argue that maybe both of these regions are either receiving input from a third region, but if they do, then that delay should be kind of comparable from the third region, or it could be that both of these regions are storing values. Each of them are storing it in their own network, so they can do the discrimination themselves. But this analysis that I'm showing you here also argues that the value signal that you see in PFC and SNR is not independent. So even though each of them may store their own version of value signal, but the value signal that is stored in them is dependent to each other. How do I show that? So for that, I show the value signal for each individual object, okay, because I show all objects to SNR and to PFC, so the objects are shared during the recording. So every dot is the AUC of one object, the value AUC of one object. Y-axis is SNR, x-axis is PFC. First of all, you can see the value signal for SNR is stronger than PFC, as most of the dots fall above the line of unity. Okay, this one is no surprise. We saw it on the firing rates. But the more important thing is that the value signal in PFC and SNR are significantly correlated. So the correlation is significant, and this is the regression line that you can see. So the value signal is correlated. These two structures are talking to each other or receiving information from some other structure. So the value signal that is stored in them for each individual object are similar. So if for one object, one region says higher value signal, the other region tends also to have higher value signal, and vice versa. And this is true for both good objects, so that here the colors, these are for good objects, and for bad objects. So for both good and bad objects, the value signal in SNR and PFC is highly correlated. And this correlation, by the way, goes away if you shuffle the label of the sets. So these are controls, basically. So someone might say, well, this is just a spurious correlation just because, some uncontrolled, let's say, things might have happened. And so one thing we can do is that if you shuffle the label of the objects shown to SNR, keep the labels the same in PFC, and redo the analysis, the correlation goes away. And plus, if you shuffle the monkey labels, so you put the SNR of one monkey against the PFC of another monkey and vice versa, right? So it's impossible that the SNR of one monkey talks with the PFC of the other monkey. There's no telepathy here. And if you do that, then also the correlation goes away. So the correlation that we see between SNR and VOPFC of the one monkey for the same object seem to be important. It seems to be correct for the same object and within the monkey that has been tested. So it is correlated. And next analysis that I did was to look at the time course of correlation. Well, we say that these firing rates are correlated, but how long does it take for this correlation to appear, right? This is time logged to the onset of the stimulus. Before the onset of the stimulus, of course, there can't be any correlation, so we are around 0. And after the onset of the stimulus, it takes about 200 to 300 milliseconds for the PFC and SNR value to become correlated. So the correlation happens around, the max is around 300 millisecond to 400 millisecond for both monkeys between PFC and SNR. And the correlation is the same for good objects, bad objects, or if you combine all objects together, which is the black curve here. Okay. So I told you I want to show you a diagram of the connectivity between these regions, and this is my diagram. And we know that cortex projects to SNR via, of course, the basal ganglia striatum system, and there are two pathways, this well-known, a direct pathway and an indirect pathway. Okay, and of course SNR then projects back through thalamus, the MDVA nucleus of thalamus, to VLPFC. Okay, and normally the studies show that normally this part of the thalamus tends to target back the same region that originated the inputs of the SNR, of that part of the SNR, and also both regions have projections to superior colliculus through inhibition and excitation, and that's why we think that the value signal that's stored in them is important for that salience that I told you about. So it's controlling the eye and gaze. But now I'm not interested in this part. I'm more interested in how these signals can be related to each other. And at this point, I had this intuition that maybe, how can I predict the firing of one region based on linear mixing of firing of another region? And, I had this kind of, the intuition comes from the fact that, if it is the case that good preferring neurons signal reach CDL-SNR via direct pathway and bad preferring neurons reach CDL-SNR via indirect pathway, then if there's this segregation, maybe we can build the CDL-SNR, explain the firing in the SNR signals. And so this is the regression we use, very simple regression. So Y bar is the average PSTH of all the neurons in one region, and I want to make it by linear mixing of PSTHs of all the neurons reported in the other region. And then I want to look at the beta coefficients and see if the beta coefficients have any relationship between the value coding of the neurons that they originated from. Okay, so I'm making this PSTH by combination of all the neurons PSTHs here, and I'll do it the other way as well. So I'll try to make PFC responses by combination of SNR responses. First thing is that if you do this regression analysis, both regions are very good at predicting the other region's response. So here I'm showing you VLPFC response, and the dashed lines is the predicted VLPFC response if I mix the SNR neurons by linear regression, very good R-squared, and I can do the opposite. I can predict the SNR responses by linear mixing of VLPFC responses, again, a respectable R-squared. But the important thing here is this. If you look at the beta coefficients for the SNR that is used to make the PFC responses, there is hardly any relationship between the beta coefficients and the AUC of the SNR neurons. On the other hand, for VLPFC, the AUC has a negative correlation, significant negative correlation with the betas have significant negative correlation with the AUC of the neuron. Okay, and if you average the beta coefficients for the bad preferring neurons with this regression, you see significantly positive betas and significantly negative betas for GP, consistent with the fact that maybe these neurons are projecting to here via direct pathway and these via indirect pathway. No such organization is seen with CDL-SNR. We don't see that. I mean, you can make these neurons, you can make these responses by combining CDL-SNR neurons, but they don't organize well with the value coding that these neurons have, and this is I think was an interesting finding. And to nail the point maybe further was that now I used 3 regressors only. So I used the average firing of all the BP neurons, all the GP neurons, and all the non-significant neurons in VLPFC, and in SN also we have 3 regressors, and I want to make the firing of SNR with only these 3 regressors. Okay, and so when you do that, the beta coefficients are like this, and the fit is like this dotted line that you see here. R-squareds are pretty good. SNR, if you just use these 3 regressors, do a pretty bad job of predicting VLPFC. Means that SNR betas, if you organize them based on the value coding of the neurons in SNR, they don't do a good job in fitting VLPFC, but not vice versa. Okay, so that's one point that we may have some clue. Of course, there needs to be anatomical studies and causal manipulation studies to prove this scheme. I'm not saying that I've proved anything, but this is an interesting correlation. So, and it's worthy of being reported. Next thing that I want to show you is the granularity of the responses. So we've recorded responses of the neurons to the amount and probability objects, okay, and the objects that have varying amount of, linearly changing relationship to the reward, and also linear relationship changing with the high reward probability. Okay, I'll show you an example here of a PFC neuron. This is the same neuron in this line. These are objects, good and bad objects. So this is a good preferring VLPFC object, and this is the response of the same neuron to objects that are linearly increasing in value. You can see the firing kind of linearly increases. I tried to color code it here, and you can see the average firing goes up, and this is the average in this window. And for probability objects, also there's an increase. So with a higher probability of a good, high reward, you get this increase in firing. So in VLPFC, you see this linear increase, and in SNR, you see the opposite, of course, right? So this is a bad preferring neuron, bad is here, good is here, and as you go linearly to higher values and of reward in amount, you get reduction of firing, and with probability you also get a reduction in firing. You may notice even in this examples that the rate of change in probability is a bit faster than in amount, and we see that in population. So if I show you the population averages for reward amount in VLPFC, we have increases here from low reward to high reward. We have decreases for SNR from low reward to high reward for amount. And for probability, we see exactly the same thing, except that you can see that it's a bit faster for probability for the increase and decrease. And if you put the firings on top of each other here, this is the top row for VLPFC, the bottom row for SNR, you see that as you increase the expected value of the objects with amount, you get increase in firing of VLPFC with amount. But then with probability, even though they're matched with expected value, you get higher firing, especially with objects that are uncertain. The objects that are certain is 100%, 0% here, they're matching, but this part is not matching, and this happens both in SNR and PFC. And if you subtract the firing of probability from amount, you get the normal expectation from uncertainty coding. It's kind of an entropy. It's a basically inverted U-shape firing difference between probability and amount, suggesting that these neurons are not only encoding value, but they're sensitive to reward uncertainty, and they code it according to the uncertainty that is maybe similar to P multiplied by 1 minus P, which is kind of the entropy of that object, entropy of the value of that object. Now, in the beginning of the talk, I told you that when I talk about salience, I always give you a behavioral measure. I don't just give you my opinion. And here I'm suggesting that uncertainty should probably be more salient. If you have matched for expected value, if an object is more uncertain, it should be probably more salient. And this is the evidence for it. That if you do free viewing with these amount objects and probability objects, the higher the amount goes. Of course, the monkey pays more attention to it, but for probability sets, you see the increase, but the increase is faster and above what you see for amounts. If you subtract the gaze bias for probability amount, you see some, again, it's not as nice and clean as neural data, but it's there that you see the added effect of uncertainty on even the behavior. So it's both in the neural data and in the behavior. This is a slide, I'll pass on it. This is about how long these two structures keep the memory. We've reported the VLPFC results previously in this paper, but in the current paper, we are comparing the longevity of the memory in PFC and SNR during time. So let's say if you train an object with reward, and then you wait hours, days, weeks, and months, and then you bring back the monkey has never seen the objects during this intervening time, and you test the neural firing in PFC and SNR, you see a retention of memories for months, which is amazing, for both VLPFC and for SNR. So this has to be sorry, the legend is switched here. This is VLPFC and this is SNR. I have to fix this. But anyways, so you see this long retention of the memory, which is important. Okay, value, I told you, has important behavioral consequences. The free viewing is one consequence, but maybe the more important consequence that, again, we've discussed previously is that if you associate value with an object for a long time, it creates efficient search. Okay, so this is, again, result with monkeys, and this is a search task. You're probably familiar with search tasks. We have target present and target absent trials, and you put multiple objects on the screen. One object is the target. In our case, the target is an object with high value, and the other all objects are low value. Of course, we don't show the monkey this red square. This is for you to know which object has higher value. So the monkey has to look at these objects and try to find the object with high value. If he finds it, he gets lots of 3 drops of reward. If he finds a bad object, he's getting 1 reward. Okay, so this is the general part of the task, and we have different group of objects, where some objects we train them before putting them in the search task for 30 days, a long time of training. It's really strong acquired salience for 5 days or for 1 day. 1 day is normally not enough to engender and engrave the acquired salience that we're talking about. So we have a gradient here, and we've previously shown, and this part of the data is published, that the search time in target present reduces as you increase the training days. So the search here is display size, the number of objects on the screen, and the time that it takes for you to find the object. It reduces as you increase the reward training prior to the search. So the search from serial becomes more parallel. Here we're also showing the target absent. In target absent, there is a reduction in also in efficient search time in rejection of the trial, but the search slope doesn't change that much. So we wanted to know what is the neural mechanism of this. And for that, my students, Kumash Sharifi and Ali Vishkai, are basically devising thinking of a hypothesis and devising a method. First, the hypothesis is this: if you're looking at a screen with multiple objects, what is one reasonable way for you to be able to find the good object quickly? I mean, one possibility is that let's say if you're looking at this fixation point here, there is a radius around the fixation that you can analyze the objects, and that's why you have to do sakat to new region to be able to analyze other regions. But what if you assume that after long-term reward training, this area expands? So this is the hypothesis. Maybe what happens with long-term reward training is that for the objects that have long-term reward experience, maybe the area that you can see them and analyze them is expanded. So you can analyze you can visually process bigger regions, where you see. And if that happens, then that can create reduction in the search slope. Okay. So the thing that Ali and Kumash have been basically doing is to try to model this behaviorally. So they're using a model, we call it multi-alternative drift diffusion process, or MAD, in which it's a very simple model. It's just, for each object we have one diffuser, and diffusion is it's just a regular drift diffusion process with the Wiener process here, some noise and some drift. And the only thing that we've added here is that a variable theta, and theta is 1 when you're looking at an object, you're rapidly accumulating evidence about whether it's good or not, and it's less than 1 when you're not looking at it. And what we're trying to see by fitting this model to behavior is to see if this theta, when you don't look at the object, does it change by reward training? Okay, so every object, so here, for example, is a 5 object display. Every object has a diffuser. If the diffuser hits the reject, you reject that object as being good, and if it hits the accept, you say, oh, that was a good object, and I'll look at it and I'll select it. Okay, so very simple model. When we fit this model, it's able to fit behavior pretty well. This is still preliminary kind of results. We're still working on it, but on the first tries, it's doing a good job in fitting target present, target absent, the slopes and search times. But more importantly, what we see as expected is that as you increase the training day, the theta for good object increases. Theta for bad objects increases a little bit, but not too much. Each color here fits with the different display sizes, and this bolder color is the mean of the displays. Okay, they're going to basically discuss the results soon in VSS. So if you're interested to see more details of this model, you can refer to that talk. Now, what I want to show you is some evidence that now we have from my lab, electrophysiology. Mujtaba Ghazizade, a postdoc in the lab, has looked at how the receptive field size might change by value. Again, we asked the monkey to look at the center of the screen. We flash objects in multiple locations, while the monkey is keeping fixating, and we're recording from VLPFC. And here, for example, you see one of the neurons, neuron number 45. The normalized responses of the neurons to good objects is here, to bad object is here. You see, for good object, you have it's as if you're responding to a bigger portion of the field, of the view field. If you plot the contours, it's a bigger contour for good objects compared to bad objects. And another example neuron is here. So it looks like really it may be true that the receptive field of the neuron enhances for objects with long-term reward value associations. And this is a quantitative measure of that and a good size of neuron population. Okay, so I think I'll skip this. This is a published study that we've done before, looking at stable value coding in the monkey brain because we don't have time, and it's kind of an older result. I'll pass by. I'll just mention that we've discovered in monkey's brain regions that hold the value memory for a long time, and predominantly these regions belong to IT cortex, the ventral bank of the SDS, and part of the VLPFC that we are recording from. So my time is almost over. We've done resting state analysis to show that these regions belong to the same network. Again, I'll pass these slides. I just want to show you one last thing is that we're now testing these things in humans. My PhD student Sepida Farmani is actually taking humans and training them with these abstract fractals for 3 days, and we do fMRI, before and after training. And we give them monetary reward for these fractals. So humans apparently are able to learn fractals, but not as good as monkeys, but they're okay, not as smart. When we ask humans to tell us whether an object that they've seen before is good or bad, their performance increases. On this first day, they're about 80-something%. Then they go above about 90% accurate, and there's some difference between good and bad objects. So they're normally more accurate for good compared to bad objects. But so this result will be presented in a poster tomorrow. But just to give you a snapshot here is that now we have about 14 subjects, and interestingly, we see intraparietal sulcus to discriminate good and bad objects in human. Also, we find some areas in fusiform, the fusiform gyrus, that does that discrimination. The locations that we find in human are different from what we find in monkeys, but especially the LIP regions, the IPS region, seem to be robust and bilateral. So it looks like there is the circuitry in humans and the homologue, even though locations in the spread of activation might be different compared to monkeys. Okay, so if I want to give you a summary of the talk, reward and reward uncertainty memory create a strong gaze bias in long-term memory. Single unit recording shows common and granular coding of value and value uncertainty memory in VLPFC and SNR. Efficient search for valuable object. We think it may arise for enlargement of processing area for good objects. Using fMRI in macaques, we found temporal prefrontal cortex and subcortical areas. I didn't show you this, but you can read it. For months, it keeps it. And in humans, we have signatures of value memory in intraparietal sulcus and fusiform gyrus. Okay, so with that, I'll stop, and I had a little bit more slides to discuss, but I don't have time, so I'll just do the acknowledgments. I want to thank lab members, the people who did the work. I mentioned them during the talk, and my collaborators in NIH, and the funders, that are basically funding our research, and the people of my lab. So some of them, part of them, is shown in this picture. So with that, I'm open for any questions.
Yes. Thank you very much. It was very interesting. My question is about the relative salience of uncertainty, and if I'm not wrong, it was value, right? That you had that you found higher salience for uncertainty relative to value.
Relative to, so it's an added uncertainty on top of value. So I cannot really compare uncertainty and value with each other because they're not on different scales. But what I find is that if an object has value, it becomes salient, and on top of the value, if it's uncertain, it has uncertainty, its salience increases based on both firing and behavioral data that I showed you. I think
it's kind of an interaction, right?
It's some sort of interaction, but it could be additive too. I'm not sure. I mean, so if you look at the behavior here, for amount objects, we have increased looking time, the red, and for probability objects, we also have increased, but this increase is more for objects that have uncertainty. So the 25%, 50%, and 75%, they have uncertainty that is added on top of the value. So both value and uncertainty cause salience.
Right.
I also showed some evidence. Yeah, here also. So these objects have the same value, expected value. These four objects are more uncertain in their value, and therefore they create more salience.
Right, and these kind of additive salience that you find, do you think this is something about the long-term history that they have, or if you make this kind of, if you change the uncertainty and value of the outcome in a trial-by-trial basis, do you expect to see something like that or not?
I have to say that we have not done the fast switchings to see how much the effect is there. But what we've done is that if you do, let's say, 3 sessions of training, and after each session you do 1 session of free viewing, the effect becomes stronger as you train more and more. So that data we've done, but trial by trial we haven't. So we haven't done an experiment where we do, let's say, value learning and then do free viewing immediately. We always done blocks of learning. So after each block there is some increased salience. And normally it kind of plateaus after 5 to 10 sessions.
Okay, thank you very much.
Yes.
Okay, I want to ask a question, Ghazizade,
Sure.
if you don't mind. Have you tried to see if that learning the value of an object has benefits in other cognitive domains? For example, in braking, if you learn something and then it's high value and it's easier to detect it in a dim condition or in lower contrast or something like that.
Those types of experiments I haven't done, but there are these types of perceptual decision-making experiments with value, let's say with ratings and stuff. Watanabe et al. Have done. We haven't tried to do it with these fractals, but with simple gratings, people have shown that if you associate simple gratings with reward, people become much better at discriminating the changes in the reward associated gratings or detection of those gratings, and that might happen subconsciously. But this is not something that we've explored.